I'd like to introduce Dr. Sean Adams, and it's actually a real pleasure to have Sean here. Sean was at the Western Health Nutrition Research Center.
That was close.
Western Human.
It's close.
Right? Nutrition Research Center on campus. It's a USDA facility affiliated with UC Davis for a number of years, which is where we got our start together doing research in obesity. He was the director of the metabolic research team at that time, and has now gone to University of Arkansas Medical School for about 4 or 5 years, and he is the director of the Arkansas Children's Nutrition Center, and where our paths cross is with obesity, and where our current very which I hope he'll get excited about today, is looking at microbiome. People know that that's kind of my interest, in looking at the microbiome and how to predict microbiome changes and health. So without further ado, I would like to introduce Sean Adams to you, and good going.
Thank you. Can I stand over here? I may. I'm really excited to be here. Thank you, Mohammed, for the invitation. I feel like I'm back at home. You'll see a lot of slides today during this talk that involve a lot of UC Davis folks, and there's a reason for that, because I was here for almost 10 years over at the Western Human Nutrition Research Center and adjunct in Department of Nutrition. And a lot of those collaborations have continued to this day. So for those of you who came in here today for answers, I'm afraid I'm going to give you a lot more questions at the end than answers. But what I'm hoping you'll take away today is that really, as we think about the GI tract and the microbiome and how diet interacts with that and how host health interacts with that, there's a whole frontier out there. There's a whole new world out there in terms of understanding how the GI tract talks to the rest of the body. And what's interesting, I hope I convince you today as well, is that this crosstalk is to really almost every organ system that you can think of. And I know in this group here, that's very important, because I know you all work on literally every organ system in the body. So again, thank you for having me today. Let's see if this works from afar. Well, everybody's heard about the microbiome. Everybody's working on the microbiome. If you haven't heard about it, you'll hear about it today. This is kind of looking at abstracts and titles of different papers in PubMed, and if you go year by year by year, and I think I got it out to 2018, you're up to something like 10,000 papers a year. Good luck trying to read all of that, right? And most of these papers, in my opinion, including some of our own, are really all about cataloging what's in your gut, okay? And we really need to understand not just what's there, but what they're doing, and I'll emphasize that in a minute. So most of the studies in this field of microbiome are really cataloging the microbiota and the microbes that are there, and then trying to correlate them with health and disease. That's sort of what we do. By the way, are there any microbiologists in the room? Or anybody with that training as an undergrad or anything? Can you please leave the room? Thank you, because I'm going to be talking about microbes and things, and I'm a physiologist. It gets ugly, so if you stay, just don't ask any questions afterward. Thank you. All right, so just a quick primer. We know that microbes in the gut, there's actually probably, kind of depends on which paper you read, there's more cells in the microbiota than there are in your body. Some people say about equal, some people say a lot more, some people say a few more. Anyway, a lot of cells, right? And what's interesting, this can be composed of 1,200 or so species at least, and we can't forget that it's not just bacteria, it's also viruses, it's also yeasts and these sorts of things as well. And those are very, very understudied in terms of their role. But they're mostly dominated by these anaerobes here. But then again, this gets even more complicated because there are species and subspecies and strains, and all these things are evolving all the time and changing their genome. This is, to me, very interesting and very relevant to today's talk. There's probably about 500 times more genomic sort of diversity, if you will, in this sort of microbiome than there is in us. So what that means is these bacteria have a huge potential for enzymatic reactions and for biology well beyond our own. And so that's going to be the crux of the talk today, trying to understand how they leverage that sort of genome, that bacterial genome, to cause differences in metabolism. What we don't know, actually, from that genomic material, we actually don't know for sure at any given moment in time, or even bioregionally in the gut, we can't be sure exactly what they're doing. We know that they have a specific capacity. So the genome tells us what capacity is there. It doesn't automatically tell us what they're actually doing at any moment in time. Here's an example of some crazy, it's a crazy slide, not to scare everybody, but basically this is to show you an example of what they can do. These are just sort of food-derived sort of phytonutrients here, and each of these letters you see here are enzymatic reactions that bacteria can do in the gut. And the reason I show this scary slide is to now imagine all of these sorts of reactions going on in the thousands, right? So not just food items that are, in your diet, but also different molecules that we are providing to the bacteria as well. And frankly, they're sort of munching on one another as well. So imagine this sort of reaction going on thousands and thousands of times, and then asking yourself, oh my gosh, what does that mean to my metabolome? How many of these end up in the blood? How many of these talk to the body? How many of these touch the liver, pancreas, the colonocytes? So these are questions we'll be starting to ask today. I guess the title says it all. These things are actually enzyme machines. So when we think of how the microbiome is impacted, there's so many things we now know impact the microbiome. And I threw a couple on here, obviously, as a pitch to the Department of Surgery, of course, right? But it's very interesting to think about some of these things medically that impact the gut microbiome. But we certainly know things like dietary fiber, phytonutrients I just showed you, different lipids, different cholesterol, bile acids, and so on. Even birth mode, C-section versus natural birth, actually impacts the microbiome pretty significantly. And there's some hypotheses that we're sort of toying around with some colleagues to consider how those changes with C-section could be talking to the brain and actually causing differences in how the brain structure looks even in infants. So that's a whole other, that's a bottle of beer sometime. Very interesting story there. But yesterday we were talking to David and Tina. We were talking about burns. I'll get to this. I added this last night because I got to thinking about one of my last slides, which is going to be related to sympathetic nervous system tone. And actually there's, I think, developing evidence of neuronal control of the microbiome. And we were talking yesterday about, wow, what happens when the sympathetic nervous system goes awry? Might that also impact the microbiome? Very interesting question there as well. All right. So here's where our sort of talk is going to be hovering today. What do these bacteria make that impact all of these different systems in the body? We know that the microbiome shifts are associated with shifts in the physiology and pathophysiology, virtually these and many, many more systems in the body. We just don't know all the signaling molecules and things that are going on, but we do know that microbiome has a profound impact on nitrogen balance and metabolism, bile acid homeostasis, nutrient availability. And these signaling molecules, immunomodulators and neuroactives, are a very, very active area of research. The serotonin pathway in the gut, for example, is highly impacted by microbiome metabolism. That's just one example. So what I'd like to ask is, cataloging the bacteria is very important. A lot of us catalog the bacteria in the microbiome, but we believe it's insufficient to just catalog them. We need to know what they're doing. And there's a variety of ways to do this, and I'll give you some examples today. You can do challenge tests, for example, a meal challenge or even a glucose tolerance test. I'll show you an example of where even a glucose tolerance test can unmask the metabolism of the bacteria that ends up with signals in the blood. So there are different ways I think we can challenge the system and understand not just the who, but the what. We've been working for a long time in metabolomics. So some of you may have heard of this sort of technology or even use it in your research or thinking about it in your practice. But it's a growing field in which essentially the metabolome is the catalog of all the metabolites in a particular sort of system. And not just cataloging them, but actually trying to understand and integrate the information from the hundreds and hundreds of metabolites, that are in the bloodstream or in the stool or in the tissue. And we believe it's kind of the downstream readout of all of these other factors here, genome, transcriptome, proteome. This metabolomics can unmask and be a hypothesis generator as to what metabolism is going on at any moment in time. We've used this for many, many years to study type 2 diabetes, insulin resistance, prediabetes, metabolically healthy and unhealthy obesity, and so on. And it occurred to us that perhaps it would be fun and very informative to leverage metabolomics to also look at the microbiome. So I kind of think of it this way. Wouldn't it be interesting to have, all of these factors here can impact the microbiome, or some folks would call it, enterotypes. We all in this room, each one of us have microbes that we sort of share, that we all kind of have, and we have a bunch of different sort of signatures of the microbiota, which differ person to person to person to person. And we think that, part of it is all these factors here, but some of you have, say, an enterotype A, an enterotype B, and an enterotype C, that if we could actually collect it and measure it, all of you would be different sitting here today. But wouldn't it be cool if enterotype A was related to metabotype A? In other words, what if you could interrogate the metabolome and actually, from the blood, for example, or the urine, and actually say, something? Somebody mentioned C. difficile earlier, right? Wouldn't it be interesting to do a blood test and look for a particular metabolite signature of the activity of that particular bacterium? Enterotype B might give you a signature for metabotype B, right? Enterotype C could give you a metabotype for C. So this is kind of the long-term goal, and it's very, very interesting to think about this and whether or not some of these things could be applied clinically in the future. So that's the general gist of it. I'm going to structure the rest of the talk, which is going to be a little data heavy, in two different ways. Really want to convince you that this is a two-way street between the microbiome and the host, this communication. The first examples I'm going to give to you today are where the diet and the microbiome shifts that occur in response to diet could influence host health and metabotype, and trying to start thinking about how some of these metabolites might actually impact host physiology. Okay? And the examples I'm going to talk about is some publications that we've done here in chronic kidney disease, and so how the microbiome can impact kidney function. And then a second example about sort of infants and being able to find metabolite signatures of breastfeeding versus formula feeding, and how that relates to the microbiome. And secondly, the second half of the talk really is looking in the opposite direction. So it's very, very interesting that host metabolic health can influence the microbiome as well, independent of diet, independent of all other factors. The host metabolic health, whether they're type 2 diabetic, whether they have increased fitness, increased weight loss, everything else being held equal, the host metabolic health somehow impacts the microbiome. And we have some ideas as to why this might be the case, but it's very, very interesting. It's going in the opposite direction. So let's jump in with a couple of examples here of how the microbiome can influence host health. Forgot. This was my pop-up. At the end, we need to ask how. At the end, we'll ask how. How do these two systems talk to one another? And to be honest with you, I don't think we really know for sure. We know about maybe a handful of molecules that we know are involved in this crosstalk, but there are hundreds and hundreds of metabolites that we haven't explored yet that I think are involved. Okay, so the first one, diet, microbiome, and xenometabolome associations with host health. We're going to talk about chronic kidney disease. And this was a collaboration done really with a great kidney physician scientist, Nick Vaziri, down at UC Irvine, working with my graduate student, Dorothy Kiefer, at the time, here at UC Davis, Oliver Fien, and others, to begin to unmask how chronic kidney disease may be influenced by a change in diet. If you think of the kidney, you start to wonder, could it be a direct target, like other tissues, of microbial signals? What about microbe effects on nitrogen metabolism? If the kidney is stressed, we know that sort of modifying nitrogen load on that kidney and sort of uremic solutes can be a very important way to improve kidney function. And then also we know that the microbiome, in fact, we started to hear a little bit today about how the microbiome can influence sort of intestinal inflammation, permeability, and those sorts of things, which can in turn impact systemic inflammation. Is it possible that by changing the microbiome in the gut, you can actually impact kidney function by reducing systemic inflammation. These were some of our questions. And I will not get into all the data. This is just an example-driven talk today. But the question was, if you give rats with a chronic kidney disease model, rats with adenine treatment here, a very high level of starch, okay, or resistant starch that feeds the lower gut bacteria. So fiber, resistant starch, different factors like that feed the lower gut bacteria and can profoundly change the microbiome. There was some evidence that potentially these sorts of shifts can actually improve kidney function, but we wanted to kind of do, if you will, the definitive test of this, obviously with a very high dose, experimental dose of this sort of resistant starch. The idea was we would shift the gut microbial populations, alter the gut milieu, and impact kidney health and mitigate, actually, chronic kidney disease. So we've published several papers here. This is just to show you that, you know, if there's interest out there of anybody who's working in kidney, come talk to me. But we've published, the first paper was really all about the pathology and some of the histology and some of the kidney function parameters, all dramatically improved actually by treatment with this resistant starch. The second paper we followed up focused primarily on the microbiome and metabolome. So trying to understand, okay, we've sort of mitigated much of the disease. What are the sort of metabolic factors that might be underlying this mitigation of the disease? And then this more recent paper here, which was done by Boris Zybulov, was very, very interesting. And it's very complicated, even for me, even though I'm a co-author. Boris does amazing work on the metaproteome. So actually interrogating the proteome of the GI tract contents. And what he finds are thousands upon thousands, as you might imagine, of proteins within that lumen and being taken out through the cecum. And 900 and some change are from the host, and thousands more are from bacteria. And so deciphering that information was very, very interesting. And he began to think about how that might impact kidney disease and nitrogen handling. So let me give you some examples of what we found. So in the GI tract, there is the production ultimately, and then in the liver, of what are called uremic solutes. And these uremic solutes can actually sort of stress the kidney, if you will. These are natural metabolites. We all have them in our blood right now. But when they're in higher concentrations, they can stress the kidney. And when the kidney has sort of has been compromised through CKD, minimizing the uremic solutes could be a strategy to try to sort of lower the stress on the kidney and improve health. So just as an example, bacteria make indole right here, as well as 4-hydroxyphenylacetic acid from tyrosine and tryptophan. So bacteria make these molecules in our gut, okay? These are converted ultimately to indoxyl sulfate, cresol sulfate, and cresol and indoxyl. These are in our bloodstream. These are handled by the kidneys, okay? So if you could modify these potentially and lower them, you might imagine that could lower the stress level on the kidneys, okay? So actually this is what we found with the feeding of the fiber here, or sort of low fiber here. Both of these models are chronic kidney disease rat models. And these are the blood concentrations of these particular molecules. So in each case, this is urine here, pardon me, urine and serum, pardon me. In each case, these uremic solutes were dropped pretty dramatically by fiber or by resistant starch feeding. So this is just an example, we think, of how diet can impact kidney function just well beyond just thinking about, you know, sort of nitrogen balance and that sort of thing. There's a colleague of mine at UAMS who's actually now following up on this by doing sort of fiber feeding and resistant starch feeding to chronic kidney disease patients and asking the question, can you improve health? So the rat studies really launched some clinical studies in this case, which I thought was an interesting example. Now this is a crazy slide, but I'm going to walk you through it. It's kind of fun, and that's why I was asking if there's a microbiologist in the room. Fortunately, there's not, I think. Anyway, what we can do is ask, well, which bacteria are involved with these different processes? One way to do that is through correlation statistics. And so when we do correlation of all of the bacteria shown here that differentiated the two treatment groups. So think about it for a minute. The two treatment groups shared a lot of bacteria, but they also have very distinct bacterial signatures. If you take just those bacteria that differentiated the two groups, and you ask, which one of how do they correlate with some of these nitrogenous end products here? For example, indole 3 acetate, indole 3 lactate, and so on and so forth. And these are from the cecum. by the way. These are cecal metabolomics. And what you can do is anything that's positive kind of looks like an orange oval going in this direction. The negative correlations are sort of the blue ovals shown here. So this is our sort of first blush, if you will, hit it over the hammer attempt to try to understand which bacteria actually associated with these changes in uremic solutes and nitrogenous metabolites. The real trick is growing these and actually diving down to make sure that the bacteria that we see a relationship with either make the metabolite or downregulate the metabolite, if you will, under these conditions. And that's a very tough type of study. So what we've tried to do is we've coupled to microbial ecologists at Purdue University who grow bacteria. They grow specific mixtures of bacteria. And what we're going to be doing is doing metabolomics on these very defined human stool-derived bacterial populations to confirm or deny some of these correlations. We'd like to know whether these bacteria actually make these molecules or not. But it's just an example of how we look at things at this stage. Another example of how the microbiome can influence host health and metabotype is in infants. And so what's very fascinating is to consider breastfeeding versus formula feeding. So this is just another example. Breastfeeding, of course, being the sort of gold standard recommendation for mothers after they've had a baby. We're studying things like, does that really matter actually? What's the science behind that? What are the differences between breastfeeding and formula feeding that confer many of the health effects of breastfeeding? So in the course of doing that, we began to ask how these two diets can influence the xenometabolome and ultimately immune function in this particular case. This study's done with a whole group of people, wonderful people at my center, but really led the way here with Aline Andres, and for the metabolism stuff, Kelly Mercer, who runs our metabolomics core lab. So the design of this study is pretty simple. It was about actually 600 children, 200 fed a soy diet, 200 breastfed, and 200 fed a dairy-based formula. Out of those, there was a subset for which sampling could occur at multiple times. So the data I'll show you in a moment are from children, very highly controlled situation, highly phenotyped through time, starting with a cohort of about 600 kids. And the question is, can you tell the difference in what a child is eating as an infant by looking at their metabolome? Okay? And this is kind of a funny way to show the data, but I think it highlights it really well. A child who was primarily milk formula fed would be shown in blue. Each of these dots represents a child. A child eating the soy diet is here in green, and breast milk in red. And what these dots represent in this 3D space is a statistical way to take all the metabolomic data from their urine, okay, hundreds and hundreds of metabolites, and mathematically determine which of these mathematically discriminate the three groups from one another. In other words, it's a predictive model called PLS-DA. And this predictive model finally winnows down the herd of hundreds of metabolites just to those metabolites that can discriminate the groups from one another. And as part of this process, each participant receives a score. And the score is a mathematical aggregate score from these discriminant variables and the variance associated with them. Okay? That score is what's plotted here. So you can see very clearly what's interesting here is that there's a clear difference in the urinary metabolome of these three diets. Can anybody see who here might have not really been in the diet they were telling us? I mean, here's the thing. I don't know that. I don't know that, right? But metabolomics, and there's a little green guy hiding in here. You see that? Really interesting. And we see this sort of thing all the time. In fact, we use this statistical approach sometimes for outlier analysis. We can use this approach and we can clearly see somebody's over here for different studies and ask, well, perhaps that person wasn't really on treatment. So that's just another value of this. So it's very, very interesting to imagine now. We can interrogate a urinary metabolome and we can really try to understand what a person is eating. And I think that's the next frontier. A lot of people working on that particular question. What was particularly interesting in this case were some of these indole metabolites. I keep coming back to the indole metabolites if you haven't noticed. And this is a very interesting pattern. So you can actually see with a huge amount of variability, obviously, differences in the urinary concentration of indole 3 acetate and indole 3 lactate in this case, with soy being high here and low here and so on and so forth. So who cares, right? Who cares? Well, what's interesting is if you actually look at the literature, there's some evidence that indole metabolites actually have anti-inflammatory effects. They have immunomodulatory effects. So we did a brief study, kind of a pilot study in my lab, where we took a pool of these indole metabolites here. Not trying to claim this is physiological, it's an experiment. If you take these indole metabolites here and you place them on a macrophage monocyte cell model, and you give them an increasing concentration shown here, the cytokine production or cytokine concentration can be dampened in these cells pretty dramatically by adding this. And what this line is right here, this is where you add endotoxin. That's a positive control. So here are cells sitting in a dish, nice and happy. Here's endotoxin, obviously causing an inflammatory response with TNF-alpha, IL-6, and so on. And these indole metabolites can blunt that effect very effectively. So we think that this is kind of a first little hint that one of the things about breastfeeding maybe could be the change in the xenometabolome, which is impacting the immune system and causing an immunomodulation of the immune system to, in response to a whole variety of things to optimize immune health. That's a big stretch, but this is what the experimental data show. Those are just some more cytokines there. So that's kind of diet microbiome to host health. These are just a couple of examples of many studies that we've done. And I'll stop here to ask, are there any questions so far or any comments so far related to some of those data or some of these questions? Yes, David.
So one question is, if it's going from the gut, it has two pathways. It goes through the liver or it goes through lymphatics. And do you have an idea which one it is? If you look at the Moore brothers studies, they suggest that for injury causing lung disease, it goes through lymphatics. So have you looked at that question?
Yeah, so we've thought a lot about the lymphatics. You know, PhDs, they think a lot. They always say a PhD will think a patient to death, right? Anyway, it's an inside joke. We don't know. So really, looking at the lymphatic metabolome is very, very interesting to us. We've never done it. We've thought about it because that's a great way to traverse different areas of the body that's not going to necessarily come through the liver or the portal system. In fact, it's now pretty well accepted that portions of the microbiome do travel through the lymphatics, actually. And that can get gut bacteria or microbes to other parts of the body that previously they couldn't understand how it got there. So I agree. I think the lymphatics are a fabulous sort of highway to get a variety of things throughout the body that we really don't appreciate yet. Haven't looked at it, though. Any other questions or comments at this point? Well, I'm going to go in the opposite direction now. I'm going to give you some examples of some very intriguing data to really suggest that the host's metabolic health somehow is impacting the microbiome. This study also, a lot of folks from Davis, including Oliver Feen, Peter Havel. Brian Picklow's been key. Brian was a UC Davis student. He now is one of the faculty members at our center. Chuck Koppel and Haney Sobey at Coppin State University. I want to highlight Haney real quick and make a point about metabolomics. One of the myths about metabolomics is, oh, well, this is no problem, especially xenometabolomics, right? You just simply measure the stuff. It's a mass spec. You just simply, and you get peaks and you do all kinds of fancy math and you're good to go. Well, it turns out there are many, many unknown metabolites that come through these systems. In fact, most of the metabolites we see, we just give a number to because we don't have a clue what metabolite they actually are. And the reason we don't have a clue is because there's no standard made. You can't just open a catalog, order the standards, and shoot it on your mass spec. So Haney has been really helpful. He's a medicinal chemist. And what Haney does is as we deduce different metabolites, like we think it may be this or we think it may be that, and I'll give you an example in a minute, Haney, thank goodness, builds it. And Haney builds it from scratch, and then we can shoot it on the mass spec, and then we can identify it for sure. So far, we've been lucky, and we've got it right in our first few metabolites, but there's going to be a lot of metabolites we don't know what they are, and it's going to be very hard to decipher what they are. All right, so Bethany, you'll appreciate this particular slide. we've begun to look at the UCD type 2 diabetic rat model developed in Peter Havel's lab on campus. And this is a very interesting rat model because it develops a type 2 diabetes-like phenotype sort of naturally, okay? And they measure blood glucose frequently, so they can actually tell animals that are pre-diabetic, newly diabetic, 3 months diabetic, 6 months diabetic, and so on and so forth. It's a really great model. Let me just give you an example. Here's insulin concentration in these animals. Here's the point of sort of diabetes onset by blood glucose measurements, fed blood glucose measurements. Insulin keeps creeping up as insulin resistance sets in, and then there's insulinopenia as the animals become sort of grossly diabetic, shown here. And this is just blood glucose level, which you can see rises pretty significantly after development of diabetes. What's cool about this model is you can actually use this model to interrogate the microbiome and the metabolome and ask the question very cleanly, does diabetes itself drive a change in the microbiome and the xenometabolome? So that's what we did here. This is showing the cecal microbiome. This is the same kind of plot I showed you earlier, where each dot is a rat, and they have a score based on their discriminating factors, okay? So shown here are early diabetic animals versus late diabetic animals, 3 and 6 months diabetic animals. And what's very unique here is not only does the microbiome appear to be different by separation of these two groups, but there's even greater separation in the cecal metabolome. So clearly the bacteria are doing something differently here. There are different bacteria as diabetes develops, but the bacteria that are there are doing something different because you're getting a different metabolome here. Very, very clearly different. This is despite the fact all of these animals were males. They were all fed the same chow, all living in the same vivarium for years. Now, the animals were not living there for years, pardon me, but the same many generations in the vivarium and so on and so forth. So we've controlled for virtually every parameter that we could think of that would alter the microbiome, and we attribute the changes really to diabetes per se. Does this happen in humans? We need to look, right, Mohammed? We need to look. We need to understand whether diabetes actually causes this in humans as well. This is another way to look at it. These are lean Sprague-Dawley rats in pink, recently diabetic here, pre-diabetic here, and in the green, 3 months diabetic. And this is a unique platform that we've developed called the Xenoscan. And the Xenoscan has now almost 200 metabolites that are from bacteria, curated from bacteria. So this is a very focused metabolomics approach, specifically interrogating the microbiome. Now imagine, and we're going to grow this, we're hoping, to many hundred. But now imagine applying this sort of technology to some of the questions I heard today that were coming up in the M&M, which I know means morbidity and mortality. Now, I know that now. Thank you. But think about it. Some of the questions that were being raised, what if we could interrogate the xenometabolome in the blood, in the sample, and so on and so forth? It may be very telling in terms of associations with disease and health. So this is a technology I just wanted to show you that we have this under development now. We're trying to get this submitted very soon. But clear separation of the groups using the Xenoscan. Now, the last few slides are really going to be all around a study I did here at UC Davis that we've been able to look at in several contexts. In essence, if we change the metabolic health in a human population, if we improve fitness, we improve weight loss, we improve insulin sensitivity, does that change the metabolome? And more interesting, does it change the xenometabolome? The answer is going to be yes, obviously, and I just want to show you some examples of how that happens. Here's the study design. These are all women. N equals about 15 at the beginning, where we had a controlled feeding period here where they didn't really change their body weight. This is percent of starting body weight here. We screen them and we do metabolomics at the beginning. So these are all obese, sedentary, insulin-resistant women, very highly phenotyped, all according to sort of accepted cutoffs for all of these different parameters. These are not healthy women coming into our study. We then subjected them to sort of a weight loss and fitness intervention with an exercise physiologist, with nutritionists, and so on and so forth. Most of them lost about, maybe 8%, maybe a little bit more body weight. And we did the exact same trial right here. And this food, all the lot numbers, everything were matched perfectly at the beginning and the end. And we asked the question, does the metabolomic pattern in these women change over time? This is to show you what happened pre and post. So clearly they lost weight, thankfully, it worked. Mohammed, you'd be proud of us. They improved their fitness level, shown here by VO2 peak, and their fat mass was significantly reduced. In addition, their insulin sensitivity was increased. So we have the same women eating essentially the same foods at both of these time points. The only difference is, in this case, they're sedentary, obese, insulin resistant. In this case, they actually have lost significant weight, they're insulin sensitive, and highly fit. And I'll show you, this is my scariest slide of the day. So I know it's early, but I'm going to wake you up with this slide here. What you can do is look at hundreds and hundreds of metabolites during any given challenge. These are metabolites during a glucose tolerance test in humans. So these are these women, glucose tolerance test, looking beyond glucose, what else changes? So we published this, and you can basically start to classify these metabolites in different groups here. For example, in lipids and fatty acids, perhaps not surprisingly, you get a glucose surge, you get an insulin surge, and the fatty acids and so on drop, as you might expect during glucose tolerance test. Many of the sugars increase, shown here in red. So blue means it drops during the GTT, red means it increases, okay? I promise you, I don't anticipate you're memorizing this, please, but I want to show you something really cool. What jumped out at me when I was looking at this ridiculous, mind-boggling spreadsheet at the time, which is basically the metabolomics of GTT, these metabolites jumped out at me right here. 5-hydroxymethyl-2-furoic acid, 2,5-furan dicarboxylic acid. Please raise your hand if you've ever heard of these metabolites in your life or saw them in your biochemistry book. If somebody raises their hand, it scares me. Okay, I don't know what they are either. So we started to do a little searching. What we found was that actually these are microbial products from the glucose tolerance test solution. The glucose tolerance test solution has different preservatives and different things that go on through the food processing. And interestingly enough, do not contain these metabolites. But it does contain this 5-hydroxymethylfuran, as many of our foods do, by the way. Many of our processed foods have that in the food. What we unmasked and what I stumbled upon here was basically that you might be able to challenge the body and unmask the xenometabolome, or what the bacteria are doing. When the bacteria saw this, they converted it, and it went up dramatically in the blood during a GTT. I just find that interesting. I don't know if it has any physiological relevance, but that was our first look into this sort of metabolic health thing. And what we found, here's another example of a xenometabolite that changed pre and post. So post-intervention is in green, pre-intervention in red. This is blood concentration of this tricarboxylic acid here. Look at the differences in pattern depending on your metabolic health. So the metabolic health of the women changed the GTT pattern of this metabolite. Who cares? This metabolite is a xenometabolite. This metabolite had only been described in ruminant cattle, believe it or not, in a couple of papers, and seems to impact things like the TCA cycle. So the fact we have it in our body is kind of interesting, and the fact that it changes pretty significantly with fitness is also very interesting. What we think this is telling us is that the microbiome is shifting in terms of its activity, depending on one's metabolic health. So the type 2 diabetes rat and this sort of study in humans, we think, really highlights this a lot. How am I doing on time? Okay. I'm going to skip that one. There's another one I want to show you, the last few slides, and I think this is super exciting, and it'll lead me to kind of tell you where we want to go next with all of this descriptive work. This is the same model. You've already seen this graph right here. But we did metabolomics during acute exercise as well. So we had these women on a sort of exercise bike at the same workload pre and post intervention, okay? And we did metabolomics analysis every 5 minutes of exercise and every 5 minutes of recovery. Another scary slide, but you guys aren't scared anymore of this kind of stuff, right? This is the metabolomics in the bloodstream in the first 15 minutes of human exercise. This paper now was just accepted on the taxi ride in. I got the text from AJP that this will be accepted, as I knew it would be. So what's interesting here is, there's a lot of stuff going on. There's some things in blue that drop during exercise, some things in orange that go up during exercise. I'm not going to get into the full interpretation. I'll let you read the paper. But What's really, really interesting is this. During 15 minutes of exercise, we began to see a variety of xenometabolites, microbially derived metabolites, changing in the blood. For example, benzoic salicylaldehyde and silicic acid here, all of these, supposedly, we don't make these. We don't make these. They either come from our food or they come from bacteria, and they go up during exercise in the blood. Chlorogenic acid is high in berries and coffee and other kinds of foods. These women were overnight fasted, guaranteed, no coffee, no tea, anything, but it went up pretty dramatically. It was one of our strongest changes in the bloodstream during exercise in humans. Dodecanol and some of these octadecanol, these are made by bacteria. These are not made by us, okay? These go up in the blood during exercise. And then finally, glycolic acid was one of our highest changes in the blood, and it's believed that this comes primarily from hydroxyproline, which is part of gelatin and cartilage. And so unless these women were breaking down their cartilage like crazy and metabolizing it, which I don't think was happening during 15 minutes of exercise, we think this comes from bacterial conversion of some of these food items. So why do I show you this? First of all, I have to admit, I'm a bit of a geek. This is just interesting. How can you get microbially derived metabolites changing during an exercise bout in humans? What does that mean physiologically? Does that mean exposure to these things and phytonutrients and other things changes acutely with exercise? We don't know. We mentioned the lymphatics in this paper. You know, are these things being harbored in the lymphatics and coming out during exercise and so on? We just don't know. We just don't know. I'll end with a couple slides on these molecules because this is, I think, very, very exciting data. Same study, metabolomics every 5 minutes of exercise, pre and post. This is following weight loss here in red, before weight loss here in black. What we found that the concentration in serum during exercise increases for this really funny molecule named cis-3,4-methylene heptanoate, or heptanoyl carnitine. Okay? So that's a head scratcher too, right? Well, I didn't learn about that in my biochemistry textbook. But let me show you what we think is going on. Here's what we think is going on. That we don't make fatty acids that have this methylene group on here, supposedly. We don't make that, okay? That can only be made by microbes. So we think that what's going on during exercise in this case is that there's bacterially derived, parent molecules here, long chain molecules here with this methylene group shown here, that they get stored in the adipose tissue as a normal part of our physiology, that these fatty acids are released during lipolysis of exercise and they get sent off to the liver and to the muscle. And then what we think is happening is a partial beta oxidation, that this long chain fatty acid is partially beta oxidized to create this medium chain sort of daughter molecule that we saw go up during exercise. So again, it's an example in our minds at least of bacterially derived metabolites that are a normal part of our physiology and actually released during exercise that we've really never thought about before. And so our hypothesis is shown here, and do we have any data to support that? So we have some unpublished data that do support this idea. Let me show you. These are brand new data, like a day before I got here. This is looking at these medium chain xeno-lipids here, cis-3,4-methylene heptanoyl carnitine and the trans version. And these are the standards here in a mass spec, LC actually, from going into the mass spec. This is muscle tissue, muscle biopsy from humans. So this is a human, not exercise, just sitting there. And you see both versions very clearly of this medium chain product. In the plasma, you actually see both of them as well. And clearly the cis version, I showed you earlier, the cis version is higher. So again, it's consistent with this idea. And now if you look at the parent molecules, remember the long chains I told you about? These are very highly abundant in feces, but not liver. In this case, we had liver from a piglet. Again, in the feces, not in the liver. Here's the medium chain version here. Very high in the liver, very low in the feces. So let me distill all this down to you. This is really, for me, very interesting, that there could be these xeno-lipids produced by bacteria, getting stored naturally in the body, we think, being released during an exercise bout or other situation, perhaps fasting, perhaps diabetes, and then being burned in the tissues to these sort of partially burned metabolites here. What we're excited about, as you might imagine, is, okay, so what, right? So what? Do these molecules have any bioactivities? We think they do. So do they have any immunomodulatory activities? Do they have any activities in some of our favorite cells like gut endocrine cells? Do they impact the liver? Do they have signaling properties? These are all studies that we have going on right now, and it's probably going to take a good 5 or 10 years to figure that one out, but it's very exciting. Okay, so one slide on mechanism. So host health impacts the microbiome. I'll admit, I have no clue how. I have no idea how. But there's one group that came out with a very interesting paper that's got me thinking a lot about the nervous system. So we know that all of our systems here somehow talk to the bacteria. We know that we feed the bacteria urea, mucus, bile acids, unknown signals, immune factors. But what's interesting is there are these other signals right here, including epinephrine and norepinephrine, which are thought now to signal to the microbiome. And this is a paper by Sewell et al., which I think is very, very interesting. Their general concept is this. These little dots here are basically norepinephrine, okay? And the thinking would be that this release of norepinephrine into the sort of GI tract lumen can have an impact on certain bacteria, commensal bacteria shown here in green, which have the ability to convert the norepinephrine to this molecule DHMA. And this DHMA, in turn, is a quorum sensing molecule, or it modifies the microbial ecology. And they have some pretty good evidence in this paper for this, that this molecule here can, in fact, in this case, attract sort of bad bacteria to this localized site in the GI tract. So, and that's shown here in red. So imagine for a moment, this is terribly exciting, because in obesity, for example, we know that the sympathetic nervous tone is disrupted. In fact, it's typical that the sympathetic nervous system is activated, chronically activated in obesity. We talked about burns yesterday. There's a situation where clearly there's a chronic activation of sympathetic tone. So it really starts you to thinking. As the nervous system is modifying signals to the gut, isn't it possible that the gut microbial ecology could change? Even if you don't necessarily change everybody who's there, you may change what they're doing, and that may be reflected in the xenometabolome. So it could have implications for health and disease, and I think this is a really exciting paper and opens up a lot of thinking in my mind. I should mention that DHMA is shown here. I think this is cecal contents. In germ-free animals, shown here, there isn't any. So these are animals with no bacteria in their gut. This molecule goes away, suggesting, of course, it's made by the gut bacteria. So, what are the take-home messages? First, there's a crosstalk, but this crosstalk's a two-way street when we think about the microbiome. The host metabolic health is critical in modifying the microbiome and what it's doing. And here's the next frontier. Where do we go? Where do we go? What does it all mean? So we know these things exist, these xenometabolites. We need to understand who makes them. We need to understand what their molecular targets are, if any. If any. For example, who makes what? What are the kinetics and fluxes in the body? Which metabolites have host and microbiome activities, bioactivities? What are the intestinal bioregional differences? What are the differences in the upper intestine versus the lower intestine? We don't know a lot of that information. And then finally, once we catalog all these metabolites and we start to think about their associations with disease and so on and so forth, and we start to even think about targets, how does that impact health and disease? How could we leverage that information to improve clinical care, biomarker analysis, and so on and so forth? Huge questions for the future, and actually keeps me pretty excited about this area, to be honest with you. So with that, I'll thank you very much and open the door to questions.
Well, thank you very much. That was really interesting, and particularly the sort of cross-pollination with so many of our different sort of surgical areas of interest. And we have a few minutes for questions.
And anybody who wants to see that slide of all the changes going on during a GTT, let me know. I'll put it back up for you. I know that David seems very excited about that. But yeah.
Thanks for the talk, Dr. Adams. It was really cool. I'll preface by referencing there was a case report. I don't think it was peer reviewed, but it was a young lady who had infectious colitis, I think C. difficile, and received a stool transplantation from another person who turned out to be a very obese patient. And this young lady was very thin. And subsequently, she got the stool transplant and did well. But very peculiarly, she started to gain a very incredible amount of weight.
Mm-hmm.
And I forgot where this was studied, which hospital, but they wrote it up and they actually isolated a couple of strains or species, or strains of Bacteroides, I think. But my question, I suppose, is, like, are you thinking of another dimension, which is, do you think that changing, like artificially changing the microbiome demographic in a host would then change the health of that host?
Of the recipient.
Yeah, of the recipient.
Yeah. I believe yes. I believe yes. Generically speaking, I think the future is bright for these sort of fecal transplant or type experiments for certain conditions. And I think there is evidence that that's the case. There's also evidence out there that in fact you can confer, an obesity phenotype from this group to this group or whatever. I think the data are strongest in rodents. There's a lot of, my understanding is, we don't do that work, but there's a lot of controversy in this area and whether you believe it or not. I don't think it's going to work in everybody. The second thing I would say about that study is that when you go from a very disrupted GI tract system to a more healthy GI tract system, you might anticipate that weight gain would occur, right? It would just, you have a healthier gut that you're going to harvest more energy naturally. So it's really complicated to deconvolute those two things, in my opinion. So I believe the answer is the recipient's health can be changed by the host's fecal sort of milieu. I just think maybe there's a little bit of overhype in the area right now for many of these different areas.
Great talk. It's obviously a very hot area, as you alluded to. We do a fair bit of work with cancer immunotherapy being applied in cancer immunotherapy as well, as you know. The issue that I think is an important one, I'd be interested to hear your thoughts, is it's very hard to really assess what's correlation versus what's causation.
That's right.
And you see that a lot in some of the studies where they, say the signature of bugs is different from, the group in France versus the group in Houston versus the group in Japan. So, what are your thoughts on how to really kind of drill down on this? Because that's really the key question for trying to apply as a therapeutic and so forth.
Right. So the question is really all about correlation and how do you go from correlation to causation, right? I would ask epidemiologists the same question, by the way. Epidemiology is generally a hypothesis-generating field, generally. I would say we're in that hypothesis-generating phase right now. So the way I always tell people, we have another maybe 5 years of kind of publishing our correlations. And we still pump out the papers and their correlations and they're interesting, but that's not going to be good enough. So what we've started to kind of go down the road of is these cultured bacteria in very defined conditions to try to understand who's actually making what. That's one of our approaches. That still doesn't get you to the physiology. So then we have another technique with the germ-free mice. So these are mouse models that have basically no gut microbiome. And you're able to do these sort of transplant of specific bacteria or populations into the recipient. And you can look for changes in their xenometabolome as well as their phenotype. So I think that's getting us dangerously close to sort of causation. But germ-free mice are weird. Yeah. So the final thing we're trying to do is we're trying to actually use I can't divulge too much, but we're trying to kind of think of it in sort of, I was in pharmaceutical R&D for many years. So you can begin to think about using cell models, specific targets, things like that in a very controlled setting, and using some of these xenometabolites to kind of interrogate those systems, and then in turn kind of going that route, which I think again gets you a little closer on the xenometabolite side to causation or involvement with a particular disease or physiology. So that's where we're going right now. You know, hopefully that'll solve some of these questions. Yeah, but it is, much of this is correlation and kind of looking at, oh, this goes up or that goes down or this, you know. So we try not to overhype. We do our best. Yeah.
So I'm going to hate to cut this conversation off because it's fabulous, and clearly you generated a lot of interest. I think I took away that we are what we eat.
Yeah, a little bit, yeah. Uh-huh.
And that you're not going to put Muhammad out of business in probably the next 2 or 3 years, but it could be soon. So
Pretty soon.
anyway, thank you so much for
Thank
coming
you, Diana.
again.
Appreciate it.